Hi everyone, welcome back to part 2. So this is part 2 of the presentation in Mullany. We just had a break. Thank you for coming back and listening. This particular part, I'm just introducing ORSAA and talking about a paper that we've published in the last year or so on genetic damage. So ORSAA was started in 2015 by a group of us who wanted to know what the science was on the topic of electromagnetic fields and health, human health, but also planetary health. The question was, is there evidence for harm? And the three of us that started it, Victor Leach, Steve Weller, and myself, we all had backgrounds where we could contribute to this. And in particular, Vic knew how to make databases, and he'd come from radiation protection. And he and Steve decided to collect as many papers as we could find and put them into a database. And Vic declared that if it was 50-50, he was going to walk away. There was no evidence, but that's not what we found. So now, after years of continually putting papers in, classifying them, categorizing them, we have over 5,000 papers. And we've always found that no matter how many papers we have there, about 2/3 of them show that there are effects. They do a study and they have a positive outcome that, yes, electromagnetic fields are having an effect. And you can go and search the ORSAA database. It's free online, and you can go and search it by topic, by endpoint, by topic, a category, in order to find out about really any particular area that you're interested in this field. We make reports from the database. We also do research and dissemination of the information and public talks like this one. So this is an example of a report that we got from the database that we put in a publication in 2022. We were just actually looking at all the different areas of health and biological outcomes that could be affected by electromagnetic fields. And these are the numbers of papers showing effects in all of these different areas. So you can see oxidative stress that was mentioned before. There we've got 79 papers, DNA damage 38, cardiovascular 11, neurotransmitters 6. There's actually more papers now, but that was in 2022. Where we wrote this paper was actually written for healthcare professionals to try and alert them to this fact that electromagnetic fields are affecting people and to try and explain why. Now, this particular paper here was written and published last year. It's based on the PhD work of Stephen Weller, and it's called a scoping review. And a review paper is when you get all the particular papers in a field and you look at them all and try and assess them and categorize them so you can actually see what's in a scoping review. Actually is a particular type of review where you actually find out what's in the field and what kind of papers are being done and what they're finding. And you usually draw graphs or images to help communicate this to your audience. And so this particular review was on genotoxicity, that is DNA damage in particular. Now, Steve read over 500 papers in detail and classified them, and then we drew graphs and reported them in this paper. The types of DNA damage that you can get from that are known, there's many different types of DNA damage, but in particular the ones that show highly in the studies that are in the database, but also in the wider worldwide database of papers. The types of damage are double strand breaks when So the DNA is, if you can imagine it, it's like a ladder. So it's got these complex sugar molecules and phosphate groups that kind of creating this ladder down the side with hydrogen bonds going across, creating strands, and amino acids, of course. Then the ladder is twisted around into a helix shape. So that's where we've got what's called the double helix, because it's got two sides and it's wrapped around and around, twisted around and around. And the types of breaks you can get often are breaks in those cross links. They're called double strand breaks when you get two of them broken in the cross links between two amino acids, or you can have a single strand break. Now, single strand breaks happen all the time with everyday stress, and double strand breaks happen with more serious stresses. Now, normally the body has a repair system that can actually, of course, go and fix those breaks, because it's essential they get fixed, because DNA is like the the recipe system for how proteins are made and how processes happen in our body. So it's really important that we don't muck around with the recipe, because if we muck around the recipe, instead of making bread, we end up with chocolate muffins, or even worse, we end up with green frog and brown snake muffins, which is damaging, not what we planned in our recipe. So, worse, really, that's my analogy, but the worst is you can end up with serious disorders, including cancer. So, the papers that in this particular review, 58% of them, there were 283 papers looking at double or single strand breaks, and 58% of them showed damage. And then there's another type of damage, DNA-based damage, where these amino acids themselves can be damaged. And that's guanine, that's cysteine. They can be damaged by oxidative stress. And 86% of 42 studies showed that type of damage. And then there's another type down here, chromosome aberrations. You can see when the chromosomes themselves start to look abnormal. They've got linkages in places they shouldn't have. And 63% of 100 studies showed that kind of damage. So you can see that there are a great proportion of studies showing different types of DNA damage. There are other types as well, but I'm just showing you the top three there. And what we did was we drew all these comparison graphs to actually explain what was going on. And this is an example of this particular one. If we looked at all of the studies all together that showed breaks, or that showed DNA damage from breaks, then we can actually see this. So there's 283 papers. And what we did was Steve broke them into in vivo, that's the green, that means live studies with where you use live animals. And then there's in vitro studies, that's the blue. There's studies where they're done in petri dishes with cells. So they're not live, and so they have problems because you're not really testing what's really happening in real life. And there's epidemiology studies. That's where you count the effects. Usually you're just counting heads, and there's fewer studies on those because particularly in this area, you want experimental studies. So within that, you can see the orange part of each section. So there's the green section showing all the in vivo studies, the live studies. And then the orange shows the percentage of papers that showed effects, and the grey shows the percentage of papers that showed no effects. And so you can see overall that of the in vivo studies, we've got 84% of them showing effects and 14% of them showing no effects. The in vitro studies, on the other hand, we've got only 39% of them showing effects and 61% of them showing no effects. So you can see from DNA breaks and fragmentation that it's more of the in vivo, that is the live studies, that are showing effects than the studies in petri dishes. And so you can we made these kind of graphs for all different aspects of DNA damage. This one in particular, so This graph here is DNA-based damage. That's the second type of damage I showed you for. You see there's a lot of studies, large percentage of those studies are in vivo live studies, and then of that, there's a large proportion of them showing effects. So that's how we represented them. And if you read the paper, you can see lots and lots of these types of graphs. But I'm just showing you the snapshots of this, the highlights of this paper. So if we looked at of the studies that did show DNA damage, what were they when we look at the different organisms and tissues? When we look at plants, insects, worms, and birds, 80% of the papers showed DNA damage. Sorry, of the papers on that topic, 80% of them showed damage. Papers on ovaries, 80% of 20 studies. Testicles, 80% of 30 studies. Sperm, 74% of 20 studies, 27 studies. Brain cells, 76% of 54 studies. Liver and lung effects, 56% of 16 studies. Oral and mucosa, 65 of 23 studies. Red blood cells, 67 of 21 studies. And there were also some studies that showed sensitivities in lymphocytes. That's in your immune cells, white blood cells. So you can see that overall, in many different aspects of tissues and animals, that there are quite a high proportion of studies showing DNA damage. And then we looked at, well, what are the characteristics of the signals that are actually showing damage? Well, we don't know purely because we've only got the papers that have been done, and there haven't been papers done on every single frequency. But if you can see here, this shows the significant DNA damage. This is the y-axis is showing the number of studies. So, for example, here in the 900 megahertz range, we've got over 100 papers, and the orange showing the percentage of those that show effects. So of the papers in that range, 65% of them are showing effects and the others aren't. Down here in the 800 megahertz range, just a little bit lower in that range, you can see it's only 50-50 of the papers there. 50% is showing effects and 51% are showing effects and 49% are showing no effects. But you can see that the papers that are, the frequency ranges that are showing the most effects in terms of the papers that have been done are in the 900, 1800 megahertz and 2450 megahertz. So this is telephone communications, and this is your modem, wireless modem in those bands. And then there's also the duration. It's not just the frequency, but then there's also how long the animals or the cells were exposed for. And studies range from less than 1 minute over to greater than 1 year. Though you can see up here, there's only 3 papers, 6 papers here. Down here, there's 19 papers. But what we noticed was that as the duration of exposure goes up, the actual percent, the proportion of effects, papers that show effects, goes down. So here we are at about 16 around. When you're going to about an exposure time of about 1 day, if you run your study where you expose your animal or your cell for about 1 day, you're going to get fewer effects than here if you're exposing the animal or the organism up for 2 weeks. There's a much, much higher proportion of effects. So it's suggesting that there's actually a duration effect. So it's not just the frequency, but the duration of exposure that's having an effect on what's going on inside the cell and inside the DNA and to the DNA. And then, as well as frequency and length of exposure, there's also intensity. And in fact, intensity is where the debate is happening. Intensity means how strong can the signal be? and standard setting bodies around the world set the level at a certain level. They say below this level, there's no harm. So we're setting a standard here. Below that, there's no harm. And if you expose above the standard, then you're out of order. So these papers actually show that it doesn't really work like that, because here we've got down here, we've got exposures at extremely low intensities. And these papers that have studied organisms themselves at extremely low intensities, 80% of them are showing effects. And in fact, as intensity goes up to very high, what we call very high, because it's above the limit. There's the ICNIRP standard that many countries around the world use, including Australia and the US and the UK, and Europe, many countries in Europe. That standard is where phones are not supposed to have a signal stronger than that, and modems are not supposed to have a signal stronger than that. Telephone communications towers are not supposed to have a signal stronger than that. If they do, they're violating the standard. But what we see, there's a couple of things about that. First of all, is that actually it's when you get up into these very high ranges that you're getting the weakest effects. Then if you get higher again and much stronger again, the effects go up again. But down here, down where you've got very, very weak exposures, you're actually getting quite a huge proportion. Here we've got a proportion of effects out of 107 papers. Here we've got just between 60 and 70% of them are showing effects. And so it's what we call a nonlinear dose response, because this is the dose in terms of intensity. And if it was linear, you'd expect as the intensity goes up, that the proportion of effects would go up like this, like my mouse is showing, that it would be kind of this linear getting stronger and stronger. But it isn't, it's going down and then up. And so the debate about where we're setting the limit, it has some problems. And the first one is that it's not a linear dose response. And the second thing is that it's been found that mobile phones, as they stand today, are actually violating this limit anyway. There's an organisation in France called PhoneGate that has discovered that many of the phones that are in use today, they violate that standard. And one of the reasons they do that is because they're tested at 5 millimeters from the head, whereas actually people put them right against the head. And the closer they are to you, then the stronger the signal. It's an inverse square law. And so that means if you double the distance, then it's 4 times weaker. So that's a problem already. So we call this the cooking standard, because it's set so very high, and yet we know there are biological effects below that limit. And this limit's been set at a point where it hits your brain by 1 degree. And it was based on rat studies on the changes in the behaviours of rats a couple of decades ago, and hasn't been changed. It's not a realistic exposure limit or standard for the types of the way that we're using technology now, which is for hours a day pressed up against the head. But anyway, what we found was here we got this nonlinear response. So we can see that here we go, this is a nonlinear response duration, and that there seems to be frequency bands that are more effective than others. So what this means is that it's not well understood. It's so complex. When you change the different parameters of the exposure, it's very complex, and we can't just rely on intensity as our guide for how biology is being affected by this technology. Now, the other thing is we know that, sorry, we'll just go back again. If you wanted to design a study where you weren't getting effects, well, you'd pick an intensity around here or around here, because they've got lower effects. You'd pick a duration around here, and you'd pick a frequency perhaps here or here, because these are the places where there are fewer effects being found. And in fact, what Steve discovered in the data was that the experimental design seems to be biased by funding, so that the other thing is that studies with cells in petri dishes are less likely to show effects. So if you run a shorter study with cells in petri dishes with certain frequencies and certain intensities, you're less likely to get effects. And it appears that there seems to be some bias going on here, because this is what it looks like. If you see orange, the percentage of papers that have effects, and these are all the design parameters of study. And here we've got the industry designed studies, and you get a similar graph for telco studies and military studies. You can see there's a lot of grey. That is, the high proportion of studies are showing no effects. And here are the studies that are run and designed by more independent institutions, government studies, university studies. And you can see they have a greater proportion of orange, that is, greater proportion of effects. So it appears that there seems to be some kind of bias going on in terms of a study design that seems to be related to who's funding the study. So that's it for the genotoxicity papers. Please have another break, and we'll come back and talk about mechanisms.